So what I would like to do is, walk you through the process related to risk-based quality management. From the past two presentations and from many other presentations that I attended, one may get the impression that it is just about the data, and you just have to link the system, the application to the data, push the button, and then you get the wonderful results, and that is risk-based quality management. But in fact, that is not necessarily the case, and therefore I would like to walk you through the process a little more in detail. And let's see how that works with the, okay. Just recently, I attended a global data management conference at Novo Nordisk. They had invited me to their conference, and I got a question at the end of my presentation about data management, and that was, they asked me, what is the greatest challenge for clinical data management? And my answer was, and obviously it was the right one, I was a little surprised, but it was the right one, to manage and understand the many systems and the many copies of the study databases. Because, we deal with a lot of data coming from the EDC system, the ePro system, and then we put them into the backend data management system. And then we create copies for risk-based quality management, and the safety data we copy into the pharmacovigilance database, and so on and so forth. So these many databases are really a challenge for many functions, not only for data management, but for many functions in the pharmaceutical and the CRO industry. So what we need to look at is, how can we get to a holistic risk-based quality management system? And just to briefly reiterate this document that probably everybody knows about, this is, the risk assessment, the risk control. It is about the risk detection, the risk history, and everything related to it. And when this process came up, people were mainly talking about risk-based monitoring. And in the industry, there was a major concern that all of a sudden we accept risks, while for the past 40 years everybody had been trained that there is no way to accept any risk in the development of a new compound. Then it slightly moved into the term risk-based quality management, because it was not necessarily only about the monitoring part. The discussion resulted in an understanding on the data management side, the biostatistics side, the project management, regulatory, medical writing, everywhere. People realized that risk-based quality management is what the right way forward would be. And we in our organization, we talk about risk-based X. So it's not only the quality management, but it could also be financing, or anything else that one can think of. Look at these data with a risk-based approach. Now, this is a graph and a process flow that I hope Steve still recognizes. I have stolen that from a DIA working group that was chaired by Steve Young. And we looked at the overall risk-based quality management process for a study. So this chart here shows the pre-study risk planning. On the left-hand upper side, you have a protocol, and you may wonder, is this the right starting point to start with a protocol, or shouldn't it be already on a therapeutic area level that one starts, or at an asset level? So this the protocol is available. Let's pretend that that is the case, and then the question, of course, is does the protocol have any design issues? Because sometimes one can already eliminate issues, risks in a study by just modifying the protocol, by, for instance, modifying the number of in- and exclusion criteria, because 95% of the patients would not meet the criteria, and that would mean a delay in the study conduct, for instance. Eventually, the risk assessment, the risk planning should result in risk assessment and categorization, based on study risks, on the risk mitigations, and eventually on a key risk indicator list. And here at the bottom of that slide, you see the critical data and process identification. Also something that I think is important to keep in mind, that we do not only talk about critical data, we also talk a lot about critical processes. And for instance, moving data from one database to another database, from EDC into the SAS environment, and from the SAS environment to the pharmacovigilance system, are processes that need to be kept under control. And I must admit, I have seen several episodes of data not making it from one system to the other, and you know, having that the authorities identify rather than the sponsor company is not a situation you want to be in. And eventually, and this is here on the right-hand side, the output should be the central monitoring plan, for instance, a central data management plan, a site monitoring plan, you know, plans that identify all the activities that the various functions are supposed to manage. And then on the second slide, and I don't want to get too much into the details here, the activities during the study conduct. During the study conduct means that the study data are being linked to the risk and to the risk indicators, and then, you know, the individual functions need to do their review. They need to decide what to do with the risk indicators when they exceed a certain threshold, accept it, or, you know, start any mitigation actions, or alert colleagues, escalate it, whatever the activity might be. I personally find these two slides very helpful, in particular for colleagues that are about to embark on risk-based quality management, because it indicates it has all the process steps available, and is a great tool to make the first steps into the risk-based quality management area. Now, when we look at the risk assessment and categorization, I think everybody is going to eventually find this document provided and developed by Transcelerate, where certain risk categories have been developed. And these risk categories help a lot to make sure that you don't overlook any potential risks in your study. Despite the fact that not all of the risk categories may be applicable to every study, but you know, it's very helpful to have sort of a checklist. And when I say checklist, I do not mean a tick box exercise list, but really a checklist where the various functions can go into these categories and identify their particular risks in the studies. And it starts with safety, you know, it goes through complexity, patient population, and goes down to geography, because sometimes, you know, depending on the type of study that you have, geography may cause a risk in your study that you do not or do get your study completed. And the categories can be expanded by your own defined areas as applicable. Now, what we did at Cyntegrity is that we piggybacked on this risk assessment and categorization tool and created sort of therapeutic area or particular risk catalogs. And as you can see here down at the bottom, there is, for instance, a COVID-19 risk catalog, one for healthy volunteers. Although, you know, one may think, you know, healthy volunteers, is it important to have a risk catalog available? Well, at least, you know, to make sure that you don't overlook any particular issues in phase 1 studies, catalogs for heart failure, etc. And on the right upper corner, you see where we are with respect to the activities when embarking on risk-based quality management. We are in the design the protocol and work with a protocol area. I think this is probably a little difficult to see, but this is part of the rect catalog, of any rect catalog, and I picked the category blinding because that is frequently being used. And then there are various numbers of questions about the blinding. For instance, how is the blinding being set up, or what is the risk of unblinding? And this is not risks that are necessarily based on data. It is risks that are based on processes, on sloppiness within the sponsor company, and not necessarily on the study sites. So this is important to keep in mind, that this risk catalog is really very, very helpful. Now, moving on in our process map to the next steps. What are the critical processes in the study? Are there any critical processes in the study? And usually that is the case. So there might be underreporting of adverse events and serious adverse events, or delays in EDC data entry, or IVRS not being followed, so misrandomization of patients. A whole bunch of processes that may go wrong, and that should be considered when setting up a risk-based quality management system. And on the other hand, there are critical data in the study. So there are data in the study like enrollment per month that are of interest, or number of serious adverse events. And we heard from the other two presentations, that you frequently see different processes and different data from the different sites, because the standard of care in France may be different compared to Canada, the United States, or Germany. So that is something that needs to be taken into account when looking at the critical data. And this also means that the study teams and project management, or whoever is involved, need to think about what is critical in my study. It's not an exercise to just push a button and create a lot of tables or graphs, because people need to fully understand what happens in their studies. So the next step then would be to go to the risk scoring. So if your risks are, for instance, here on the left-hand side, enrollment or injection volume or comparability, or that one of the in and exclusion criteria might be the study requires 100 to 200 male patients, then the impact, the likelihood, and the detectability needs to be determined. And whether you use a 3-step scale or a 5-step scale doesn't make too much a difference. At least in the end, you create sort of a risk score. Now, the risk score does not necessarily mean that the higher the number, the greater the risk. It could be that the impact is really huge, but the likelihood and detectability, likelihood is low and detectability is without any problems. And still you may want to assess that risk because it is too important for the study, and you don't want to take a chance that a particular, like the 100 to 200 male patients required, that one is being missed in the study. Then, of course, it's important to have mitigation actions in place. So this is just an example using the same risks on one hand, and a couple of mitigation actions that one can come up with to ensure that the moment that the risks materialize, that you have activities, mitigation activities, actions in place that you could apply and make sure that the risk hopefully disappears again. And at the end of the exercise, and this is just the Cyntegrity way of displaying it, you have all the risks in for a particular trial, and you have the color code, you have the risk indicators, color code indicating there are red and the yellow and the green ones, and you see that from one study to another, it may really differ depending on, for instance, the sites that you want to participate in the trial. When taking a look at the entire process from the protocol development here on the left-hand side over to the site activation, patient recruitment, treatment, study management, patient discharge, and eventually study reporting, you have a lot of areas where you have risks and key risk indicators that you need to measure. So for instance, first patient in and the inclusion criteria during the patient recruitment, or during study management, data management activities, compliance and monitoring, or the safety management. So it is important to keep in mind that this is a holistic view and a holistic problem that needs to be looked at, and not just individual cells in isolation. Now, what could be the sources of the data? And that is something that the colleague from ThoughtSphere and also from CluePoints indicated already. The source of the data is not only EDC. That is something that happened in the beginning. Everybody thought my data are in the EDC system, and therefore that is what I need to tap into. But in reality, the data come also from the IVRS system, CTMS, finance, investigator data, pharmacovigilance, could be the lab information systems or other third-party sources, and we heard about that, patient diaries, etc. So it's important to keep in mind the risk-based quality management does not only focus on EDC data, because EDC covers maybe 60%, maybe 70% of the data, but there are lots of other data that are important to keep in mind. What many companies struggle with is the frequency of the KRI measurements. So should we run the analysis every day, or should we run it every month? And I think the critical exercise is to understand the study. And Steve just mentioned, a study that they handled with a vaccine. And my understanding of vaccine studies is that usually a lot of patients are being treated in a very short time. So the frequency of the analyses must happen probably on a daily, maybe every second day basis. While, in a rare disease case, the frequency may be just once per month, because there are only 2 patients enrolled per month, and therefore it doesn't make sense to have the analysis done every day. And then, of course, once you have, again, your risks, you need to find the risk indicators for your risk. And just to use one of the examples here, enrollment, the indicator could be the number of subjects per month, or the number of subjects per site, or another indicator could be the length of the break in September in China, because all of a sudden China takes 4 weeks off, not 4 years, 4 weeks off, and all of a sudden the enrollment is almost completely stopped. So that is something that someone may want to keep in mind and look at and measure via the key risk indicators. So then the risk indicators need certain thresholds. For instance, thresholds that trigger monitoring activities, or review the extremes for plausible explanations. So down here you see a risk indicator overview. It's lots of green bars, so be happy. But there are yellow ones and there are red ones, and there are red ones that show up and disappear again, and show up and disappear again. That is, for instance, the over-reporting of serious adverse events. That is not an issue. That is sort of a random issue that happens, and obviously has been corrected by whatever means. But these 3 areas here, those need to be looked at much more closely, and eventually someone, could be a central monitor, could be the monitor at the site, should decide what to do with respect to the high number of follow-up visits, or the high number of I can't read even of treatment visits that are missing, obviously. So that is something that someone needs to look at and create and start any actions to prevent this from happening again, or to continue like here in this display. And finally, almost finally, we need to look at trends and patterns and outliers and predictions. So trends and patterns, and we saw those in Steve's presentation. with the graphs and the outliers and the extreme values, that is, of course, something that someone needs to look at, in particular in the centralized monitoring system. But what is also interesting, and this is something that I personally always wished I had available while I was working in data management at Bayer, sort of the predictions. So what is going to happen to my data? And you may have, for instance, here consented subjects in January, or you look at the consented subjects already in November. But you can run these predictions not only for enrollment, but you can run this also for serious adverse event reporting or any other indicator that you have. And that is something that I personally think is very helpful to have those predictions in place. And then, of course, centralized statistical monitoring. We heard about that earlier today. So look for digit preferences. And we see that rather frequently, that when you look at blood pressure values, they usually end up with a 5 or a 0. And you wonder, why is that? Well, this is sort of an individual rounding factor that is being applied. Or you look at the heart rate, and the heart rate always ends with an even number. And why is that? Despite the fact that the protocol says you are supposed to measure the heart rate for 1 minute, site staff doesn't do that. Investigators don't do it. And they measure it for 15 seconds and then multiply it by 4. And all of a sudden, we only see certain numbers, in particular even numbers in the data. So there is a whole bunch of things that you can do to look at the data, find outliers, and then dig deeper into the data and find the root cause for it eventually. So in conclusion, when implementing risk-based quality management, one has to consider analyzing the data on an ongoing basis once they were made available. On an ongoing basis, as I indicated, means one has to look at the frequency and not do it too frequent, but frequently enough to be able to identify the risk. And then secondly, I think it's important to assess all the steps necessary to create a holistic risk-based quality management environment, because nobody really on the client side, on the customer side, is interested in getting a system that you have to leave and go back into again, and for different activities like the risk assessment and the critical data and critical process determination, you go into one system, but then you have to move to another system to look at the output of your data. Preferably everything that I listed here under bullet point number 1 to 15 should be covered by one system. And this is probably not even a comprehensive list. I'm pretty sure colleagues have more ideas on what else could be added. And what is important and very, very close to my heart is do not copy the data every time you want to run an analysis. Copying data these days becomes more and more difficult, and preferably one just links to the data that are sitting somewhere and runs the analysis and works with the results, but leave the data where they are. Do not copy the data. And once such an approach has been implemented, I'm pretty sure that users shall be able to manage all the risk-based quality management related activities within such a system, and get used to working with an RBQM environment.